Hello and welcome to my next video. Today I'll be talking about a new cancer treatment that uses nanomachines that can specifically attack cancer cells and not affect normal cells. This nanomachine has been designed to change its conformation based on what part of the cell it's interacting with, and this allows it to enter cells without needing to actually damage the cell on its way in. And it also is able to selectively enter cancer cells and ignore normal cells. So I'll get into some of the technical aspects of it, kind of explain, what was protecting a cell normally and why cancer cells are able to be attacked specifically, and also what the nanomachine does when it enters the cell and sort of how they plan to use this for actual cancer treatment. So starting with a basic diagram of an animal cell, you can see there's a lot of things going on, a lot of different what are called organelles in the cell, but what we're really going to be focused on is this outer membrane called just the cell membrane. And it has a few unique aspects to it that protect the cell from things that might be trying to enter or damage the cell. And a lot of it comes down to the structure called the phospholipid bilayer. Maybe a complicated name, but essentially means it has these molecules that have a sort of a head and a tail, and then there's two layers forming a bilayer where the two tails are facing each other. And what this allows it to do is have a hydrophilic side on top and bottom of the membrane, and then the inside of that membrane is hydrophobic or lipophilic. And these words basically mean that the outer part can interact with water and the inner part can interact with oils, and they'll block each other. So normally the way that things enter cells is that they have these specific parts of the cell that sort of intentionally bring things in or push things out. So in order to enter the cell without going through these channels and actually go through this bilayer, something would need to be able to interact with water and oils and be what's called amphiphilic, basically meaning hydrophilic and lipophilic at the same time. And that's actually one of the things that they've designed into this nanomachine, is that it's able to sort of change its formation to allow it to interact with water and then switch to interact with oil. So here in the first part of the figure you see one of the arms that's on this nanomachine, and it has a pretty cool design where the arm sort of has different segments that are able to interact with different things, and it sort of extends the arm if it wants to interact with the hydrophobic part of the cell, and then folds the arm back in and exposes that green part that can then dissolve in water and become hydrophilic again. And really the key part of this is that it's a very simple machine, only consisting of a few molecules, but it's able to automatically change its conformation. As you see the second part of the figure, it approaches the membrane being dissolved in water. Then once it starts interacting with the membrane, it automatically extends the arm, enters the oil phase of this bilayer, and then goes through the layer, and then leaves and dissolves into water once again. Then once it successfully enters the cell will break down the nanomachine and expose the particle that's inside of it. So now that the nanomachine is able to peacefully enter the cell, there's the question of differentiating between normal cells and cancer cells, since presumably this would be killing the cells once the nanomachine enters. You want to avoid killing normal cells and only really target the cancer cells. And it's actually done by adding another molecule to the system that is sensitive to the pH level or the acidity of the area around the cells, essentially. You can see in this figure that a normal cell has a pH around it of about 7.4, which is basic, versus a cancer cell, which would be acidic with a pH below 7. And when the environment is basic for a normal cell, the additional molecule will be in this red form that allows it to stick to the nanomachine and prevent it from actually absorbing into the membrane. Sort of locks it and stops it from extending these arms that it has. Whereas in a cancer cell, the pH is lower and it changes this molecule into the green state, where it has basically the same charge as the nanomachine, and it doesn't associate with it anymore and doesn't block it from changing the position of the arms. And this really is a quite elegant solution, since obviously normal cells and cancer cells are actually pretty similar in their actual structure. They both have the same sort of bilayer membrane, but this one difference of the slight change in pH allows them to only attack cancer cells and sort of make the nanomachine inert for normal cells. So the last question is, what does this nanomachine actually do once it enters the cell? And it simply just sort of gets dissolved by the cell. The cell is really good at dealing with things that enter it that aren't expected. But the issue is that it actually contains a gold nanoparticle. You might have seen the figure earlier where sort of has this yellow center to it. It doesn't seem like it would do that much to the cell, but it actually triggers a sequence called apoptosis, where the cell basically has a programmed way of dying once it's usually once the cell is too old or is damaged in some way. But this gold particle sort of prematurely triggers this process, and this is a good thing, obviously, in cancer cells where the cells are growing out of control, and this gold particle entering it would just, kill the cells and stop them from growing. Anyway, this seems like a pretty promising new technology. Assuming they can really confirm that the nanomachines have no effect on normal cells, to actually allow them to just, basically just inject these nanomachines into somebody that has, a suitable type of cancer, and the nanomachines would basically just find their way to the cancer cell and selectively destroy them. But obviously this isn't going to be the new cure to cancer necessarily. There's probably still a lot of work to be done. They haven't even started clinical trials or really animal experiments with this, but this is very promising, and hopefully we'll see more from this lab and see if they can do something else with this technology. So as usual, I'll leave a link to the article in the description, and I guess if you found this video interesting, like, comment, subscribe, and I'll see you next time.